Welcome back to my course Aspects of Biochemical Engineering. In last couple of lectures, I try to concentrate on two different aspects. One is that chemical reaction kinetics and another is the reactor analysis. And since that biochemical engineering and chemical engineering, they are related, so first we should know the ABC of this chemical engineering, particularly reaction kinetics and the reactor analysis. Now, with this information, now we will enter into the real topic, and today the topic that I am going to discuss, that is kinetics of enzyme catalyzed reaction using free enzymes. Now, as you know that in the microbial system, if you comprises of multi-enzyme system, different enzymes, they have metabolic pathways. In the metabolic pathways, your substrate is degraded to different products. Now, if you look at the metabolic pathways, the different steps, individual steps, they required individual enzymes. So, basically we considered the microbial system as a multi-enzyme system. So, understand the microbial system better, first we should know what is enzymatic reaction kinetics. So, this present lecture and couple of lectures I shall concentrate with this, try to understand what we mean by enzymatic reaction kinetics. And first we shall have to know that what is called enzymes. Now, if you look at the enzymes, is the globular protein that acts as a biological catalyst. Now, what do you mean by globular protein? If you look at that, the structure of the protein, it is of two types. One is globular protein and there is fibrous protein. Now, what is globular protein? It is randomly folded. This is like this. It is randomly folded, and when it folds randomly, some active site might be developed. Maybe this active site is developed, and this configuration of this active site is similar to the structure of a particular substrate molecule. This is the enzyme and this is this. So, then this substrate sit here at the active site and react to give the product. So, but fibrous protein is not a fibrous protein, this is kind of structure like this. So, they have now this folded structure of the globular protein and this fibrous structure, this is mostly due to the hydrogen bond, and this hydrogen bond in between the R group, we know that 20 different amino acids are there. R group of 20 different amino acids in combination with amino and carboxylic acid give, they make a folded structure. So, this is how they form the active site. Now, the role of the enzyme is a catalyst, and as you know, the catalyst role is they take part in the reaction and they reduce the activation energy of the reaction, so that rate of reaction is increases, and after the reaction is over, it remain unaltered. So, one specific feature of the enzymatic reaction is the enzymes are very specific with respect to substrate. Another thing I forgot to mention, that as I told you that enzymes are globular protein, but not necessarily enzymes are protein. It may be some pure organic molecules, and due to advancement of organic chemistry, now it is possible to produce some kind of synthetic enzyme. I can give the example of RNAs. So, another very important thing is that what you call the nomenclature of the enzyme. Nomenclature means how we name the enzymes. Most of the enzymes you usually name ended by S, as for example, lipase, amylase, protease, like this. But there is some exceptional case where some enzymes name is pepsin as you know, pepsin is remaining in our stomach. This is rennin. It is largely used in the cheese making industry, and trypsin that is present in our intestine. So we have the usually the name of the enzyme is ended by S. Now enzymes may be of two types. One is called endoenzymes and other is exoenzymes. The endoenzymes means they work within the cells. As for example, if you look at the enzyme pathway, we have so many enzymes participating in the reaction, and most of the reactions are their endoenzymes. And all but exoenzymes is basically it is a hydrolytic enzyme. The organism this produces and it comes out of the cells and then interact with the substrate, and substrate will undergo the hydrolysis and give the product. So this is the examples I can give you. The enzymes that function within the cells, the metabolic enzymes like cytochrome oxidase. Then I can give the another example of phosphofructokinase of the enzyme pathway. Enzymes catalyze reaction outside the cells. We have different hydrolytic enzymes like amylase, lipase and protease. Now I was telling about that how enzyme participate in the reaction. Now enzymes, it lowered the activation energy. If you look at this figure, that normal enzymatic reaction, you require this much of activation energy. This is the normal activation energy that is required. Now when the presence of catalyst enzyme is there, the activation energy is requirement. So this is much less as compared to EA. So since this activation energy requirement is very less, so if enzymes present in the reaction mixture is accelerate the rate of reaction. The only thing is that after the reaction is over, the enzyme remains unaltered. But one thing I want to highlight here, it does not affect the reaction equilibrium. Now whatever enzymes we have, broadly it can be classified into 6 different types. As for example, we have oxidoreductase, this is one enzyme, then transferase, another enzyme, then hydrolase, that is another enzyme, then lyase, then isomerase, then ligase. So all the enzymes more or less they fall under this category. Now oxidoreductase, the enzymes which take part in the oxidation reduction reaction. What do you mean by oxidation reaction? You take out one electron, and reduction reaction will add in the electron to the molecules. The single replacement of this, you can see that this is replacement, that how it is taking place here. This is transferase. Hydrolase means this is water molecule, this is the substrate, and when it hydrolyzes, it produces 2 compounds. I can give the example of sugar. When sugar hydrolyzes in presence of water, it produces glucose and fructose. Now another enzyme is called lyase. This undergo the decomposition. I have given a lot of examples here. The decarboxylase, hydrolase. This is how the reaction takes. Isomer is very interesting enzyme which change the functional group. As for example, glucose is converted to fructose, and glucose has the aldehyde group and fructose has ketone group. So naturally that for this change transformation we required one enzyme what we call glucose isomerase enzyme. Now this is called isomerase. Then we have ligases. Ligases means 2 molecules they attach together. You can see this. This is how enzyme classification can be done. Now let me tell you that what are the different components present in the normal enzymes. So enzymes has 2 components. One is called apoenzyme. One is called apoenzymes and another is the cofactors. Now, what do you mean by apoenzyme? Apoenzymes is the kind of, we consider kind of it is the protein molecules, but this is inactive form. But the apoenzymes, I told you this is inactive enzyme, this is not active. So it will not react with the substrate and give the product. But in presence of cofactor, now cofactor may be of two types. One is inorganic component, when we have manganese ion or magnesium ion, or you have zinc ion. And coenzymes, this is non-protein organic molecule like NADH, FADH, ATP. So those molecules we consider as a so coenzyme, basically this is the organic molecule, and some other than organic molecule we consider also the cofactor or inorganic molecule. Now, apoenzyme plus cofactor or coenzymes we call holoenzyme. Holoenzyme means active enzyme. Now, I can this is pictorially can be represented like this. Now, if you look at the configuration of the substrate, this does not have any resemblance, much of resemblance with the configuration of the active site. As soon as that your coenzyme sits here, then you can find it has a resemblance with the substrate. The substrate can sit at the active site and give the product. This is how apoenzyme is activated. Now, question comes how this active site formation take place. This is very interesting. As you know that we have 20 different amino acids. Out of 20 different amino acids, R group of 8 different amino acids like aspartic acid, cysteine, glutamine, histidine, then lysine, methionine, serine, and threonine. And this in combination with the amino and carboxylic acid group, they form this active site. They form this active site. Then substrate sit here. This is so here I want to highlight only one thing, that protein with active site we call it enzyme, and protein which does not have active site we cannot call it enzyme, because enzyme should react with the substrate and give the product. Now, question come how substrate and enzyme they interact with each other. There are 3 different hypotheses that we have that can be described, that how substrate and enzyme they can interact with each other. First hypothesis is the lock and key hypothesis. What is lock and key hypothesis? As you know that when you open a lock, the key should be proper. The lever we have in the lock, we have lever system, and if your key does not fit in the lock, then you cannot open it. So similar to the lock and key hypothesis means the active site of the enzyme should be the ditto. This is the same as the configuration of the substrate, so that you can sit at the active site. If you sit properly at the active site, then you will give the product. Now, I can give an example here. You can see that it is pictorially demonstrated here, that suppose this is the enzyme and this is the substrate, and this has resemblance with the configuration of the substrate has resemblance with the configuration of the active sites. So, substrate can sit here and give the product. Now, other two the hypotheses are, one is called proximity effect. What do you mean by proximity effect? Proximity effect means when substrate approach to the active site, because if apparently when substrate you look at the configuration of the substrate and configuration of the active site, they are not resemblance with each other. But as soon as the substrate approach to the active site, it due to induction there will be change in the configuration of the active site, so that your substrate can sit at the active site properly and give the product. I can give a very simple example here. You look at here that this is the substrate molecule, let us assume that, and this is the active site of the enzyme. So, we can easily find out the configuration of the substrate and configuration of the active site, they are not same. But as soon as it approaches here, the configuration changes. so the substrate molecule can sit properly and give the product. Now, another is the orientation effect. What do you mean by orientation effect? Orientation effect means, suppose that when substrate, so you know that enzyme, I told you that enzyme with active sites, we call it protein with active site, we call it enzyme. Now, suppose how the protein formation take place, due to polymer of amino acids. And I told you when I previously that if the number of amino acid in between 20 to 50, then we call it peptide, and more than 50 amino acid we call it protein. The protein, now that is the polymer of amino acids. Now, what is happening that a tiny part, a small part of the protein molecule, that is the active sites. So, suppose the active site, it is your substrate is this side and active site is the other side. So, as soon as your substrate approaches to the enzyme, then some orientation take place of the enzyme molecules, so that substrate can sit properly at the active sites of the enzyme. So, this is the orientation effect, and this is also taking place due to induction. Now, I told you that enzymes are very specific as per activity is concerned. So, there are this specificity we can divide it into 6 different categories. One is called bond specificity, and there is group specificity. One is bond specificity, another group specificity, substrate specificity, optical stereo specificity, geometrical specificity, and the cofactor specificity. I have given different examples here. As for example, amylase can hydrolyze alpha 1,4 glycosidic linkage, because you might be knowing that alpha amylase act on the starch molecule. What is starch? Starch is a polymer of glucose which are bind together with the help of alpha 1,4 linkage. Now, group specificity, that trypsin hydrolyze the peptide bond provided by the arginine and lysine. Then substrate specificity, the lactase acts on lactose. I have given the example of another substrate specificity like glucose isomerase act on glucose. It cannot act on lactose, fructose. The optical or stereo specificity means L amino oxidase act only on L amino acids. So, we know that two type of optical isomers is the L and D. So, L amino oxidase can act on L amino acid. What is geometrical specificity? That the alcohol dehydrogenase can oxidize ethanol, methanol, and propanol. Cofactor specificity means DNA polymerase is inactivate in presence of magnesium ion. Now, next point that is very important, what you call enzymatic activities. How good and bad is the enzyme, that can be expressed with the help of activity. Now, activity can be expressed in different ways. One is called IU. IU means international unit. What is IU? The micromoles of substrate degraded or micromoles of product form per milliliter of enzyme solution per minute. This is decided by the IUPAC, that is the international body. They decided the definition of IU. Now, if we express the enzymatic activity is 5 IU, 2 IU, 3 IU, then we shall have to we don't have to explain what do you mean by IU, because this IU has been defined by the IUPAC. But if you express in the unit that is not IU, then you have to define. This may be in different way, as I have given one example, that either micromoles or microgram or milligram of substrate degraded per milliliter of enzyme per minute. You can explain whatever things you like. Now, another activity that is called specific activity. What is specific activity? Micromoles of substrate degraded per milligram of protein per minute. Now, here I want to stress one thing. When you go for the specific activity, that implies that characteristics of the enzyme. That means that enzymes are good or bad. Actually, that we can easily find out with the help of specific activity. But if you IU, that it may vary, because if you dilute the enzyme solution, then IU may vary. But specific activity will remain constant. This determine the characteristics of the enzyme. Another is very important thing is the turnover number. Turnover number, when the protein molecule is very big, it might, there is a possibility it might be having more than one active site. In that case, the activity is expressed moles of substrate degraded per active site of the enzyme per minute, and Kcat equal to Vmax by E0. This is how we can express that. Now, Michaelis-Menten that propose one equation. This equation is V equal to Vmax S Km plus S. Now, this equation is purely on the basis of the correlation it is shown here. And this kind of correlation, if the correlation is this, and this Michaelis-Menten equation is not valid. Now, what does it tell? It tells at low substrate concentration, when you are here, then V it tends to Vmax. How V tends to Vmax? At low substrate concentration, then what is happening that if you this is low, then I can ignore as compared to this. Then this is constant, the V will be equal to constant into S, and this is first order reaction. So, it follows the first order kinetics. Now, in case S is very high, then we can ignore that, then SS will cancel, then I can like V tends to Vmax. So, this is exactly happen here, where V this is then what is the reaction? Reaction followed a zero order kinetics, because V will be equal to K into S to the power 0. So, zero order kinetics, and maximum velocity of reaction is proportional to the initial enzyme concentration. Now, Michaelis-Menten proposed this equation purely on the basis of correlation between velocity of reaction and substrate concentration. Later on, the Briggs and Helden, they proposed the enzymatic reaction kinetics to justify Michaelis-Menten equation. What they told, the enzyme and substrate, they reversibly react and form ES, and when ES form, then it produce enzyme and product. Now, what do you mean by that? Suppose I told you that when your substrate approaches to the active sites of the enzyme, is supposed to sit at the active site. So, this sitting phenomena is maybe like this, some sit properly, some split, some can slip like this. So, this phenomena we consider as a reversible phenomena. This is cannot be irreversible. But whatever enzyme, whatever substrate sits at the active sites of the enzyme, that give the product. So, that is irreversible reaction. This is what Briggs and Helden they propose. And this is how the reaction can be expressed. This is E plus S equal to E S and E like this. Now, rate of the product formation. So, what is our equation that we have? E plus S reversibly give ES, am I right? And this gives irreversibly, it gives the E plus P. So, this is K1 and this is K minus 1 and this is K2. The rate of product formation here, this is plus P, am I right? Rate of product formation, this is different. Rate of product formation is what? That K2 into ES. This is K2 into ES. But rate of formation of ES is proportional to, this is forward reaction, this is backward reaction, again this is degraded. So, it is K1 into E into S and K minus 1 ES, K2 into ES. So, now one thing we shall have to remember, enzyme concentration any point of time, E0 is the initial enzyme concentration equal to free enzyme plus bound enzyme. This is the so EI concentration of E I can express that E0 minus ES. Now, when the Briggs and Hellem try to justify this equation, they have certain assumption that what they are saying, when you plot here, you see here plotted S by S0 versus time. When this plot is like this, then your this quasi steady state condition is valid, and at the quasi steady state condition, dEs by dt, this should be equal to 0. But if the relationship is like this, what is shown here, then your Michaelis-Menten equation will not, that quasi steady state condition is not valid. Now, what do you mean by quasi steady state condition? Quasi steady state condition means tends to steady state. It is not exactly steady state, but the situation is going to the steady. That is called quasi steady state. Now, this is finally they have derived this equation, and this is simple. I hope you can all calculate this. This is shown here how it is done. And what I want to point out, this value k minus 1 plus k2 by k1, this is equal to km, and and another thing that we have k2 into Es, that is called Vmax. This is basically this is called Vmax. So I can write like this. So this equation can be easily write in this form. This is how we can write in this form, and this is where Vmax equal to k2 into E0 and km k minus 1 by like this. Now question comes, how we can what is the significance of the value of km and Vmax? Because if you look at this equation, Vmax is km plus S, am I right? So what I can tell that V is directly proportional to Vmax. So as the Vmax value increases, velocity of reaction increases, and V is inversely proportional to Km. As Km increases, velocity of reaction decreases. So here what I have pointed out, Km is equal to the substrate concentration, because we have seen this is V versus S plot, it is like this. Now here suppose this is the Vmax and this is Vmax by 2. Now if you put this, then you will find this is equal to Km value. This is the if you V equal to Vmax by 2, if you put in this Michaelis-Menten equation, then you will find that S equal to Km. Now higher the value of Km, lower the substrate affinity. This is exactly how it is proportional, and lower the value of Km, higher the substrate affinity, that is more velocity of the reaction. So what does it mean? They mean that suppose I want to produce a certain amount of product. For if the Km value is low, we require less amount of substrate, but if the Km value is high, we require more amount of substrate. That is the significance of this. Now question comes, how we can determine the Km and Vmax value. There are three different plots we have. One is Lineweaver-Bach plot, another Adie-Hupsey plot, and Hens-Henul plot. And these plots are very simple. That is, the Lineweaver-Bach plot is considered as the inverse relationship. Like we have Michaelis-Menten equation, V equal to Vmax S Km plus S. Am I right? Now if you just inverse, you will get this relationship. Just you inverse that, you will get this. So here you can see this is y, this is 1 by v, we can write y equal to, this is constant, Vmax is constant C, and this is also constant, this I m into, and this is I can constant m into x. So y equal to mx plus c. So it is the straight line plot. So if we have this slope here, and this will give you the value of Km by Vmax, and intercept here will give you the 1 by Vmax value. Now similarly, we can see that Adie-Hupsey plot, I can write this Michaelis-Menten equation in the form of this form we can write. Now if you write this form, then V, if you plot V versus V by S, then what will, this is negative slope. So the negative slope will be there. The slope will be, if you take the slope, those slope will be minus Km, and this intercept, if you look at this this is V, so this maximum value we can consider as the Vmax. So directly we can we can find out the Km and Vmax value. Now, Henson-Wolf plot we can write in other way. If you look at this is like this, S by V equal to Km by Vmax 1 by Vmax into S. So, it is something similar to Lineweaver-Bach plot. It is also straight line plot. Only the thing is that the slope here slope will give you the value of 1 by Vmax, and intercept will give you the Vmax by, because what is reverse in case of your Lineweaver-Bach plot. So, question comes, what are the limitations of the Michaelis-Menten equation? There are several limitations. I told you when I discussed the chemical reaction kinetics, when you write any kind of mathematical equation, we make a certain assumption, and all the assumptions are the limitation of this equation. I have given the simple example of that ideal gas law. Ideal gas, what is the ideal gas law? PV equal to nRT. And what is the limitation of this equation? Because this is valid for the ideal gas. And what do you mean by ideal gas? Ideal gas means it is imaginary gas. This is non-existence. So, this is one thing you should remember. Another thing is that this Michaelis-Menten equation does not take care of the inhibition of the it does not take care of the inhibition that that take care inhibition. And another important thing is the Michaelis-Menten equation considered one substrate, one enzyme. Because if you look at the enzymatic reaction, most of the enzymatic reaction they required the cofactor. So, basically it is two substrate and one enzyme molecule. So, in that way this does not explain that two substrate and one enzyme. So, these are the limitations of this Michaelis-Menten equation. So, in conclusion, I want to tell that I try to explain you that what do you mean by enzymes. I told you enzymes are basically they are protein molecules. What is protein? Protein is nothing but polymer of amino acids, and protein has two different structure. One is called globular structure, another is fibrous structure. That protein with globular structure, they are responsible for the formation of the active sites, and so protein with fibrous structure they are not enzyme because they cannot form the active sites. Now, the two type of enzymes we have, one is the endo enzymes and the exo enzymes. Some enzymes that work inside the cell, some enzymes go out of the cell and work outside out of the cells. So, so then I told you whatever enzymes are there, it can be classified into 6 different types. And then I also explained the how the substrate, what are the 3 different hypotheses, how substrate and enzyme they can interact with each other and give the product. And finally, I discussed the Michaelis-Menten equation, how Michaelis-Menten proposed the equation for rate of the enzymatic reaction. And then later on Biggs and Helem, they came forward to justify the Michaelis-Menten equation with the help of reaction kinetics. And I try to discuss what are the limitations of the Michaelis-Menten equation, and I also discuss what is the significance of the reaction constants Km and Vmax value, and also I also conclude that under the kind of limitations of this equation, in addition to the what are the assumption we have made for the Michaelis to justify the Michaelis-Menten equation. So that is all for this lecture. I think in the coming lecture I shall discuss about the inhibition of the enzymatic reaction, which has lot of application in the biochemical industry. Thank you.